Hello and welcome to this video presentation on our research. I'm Dr. Aidan Mead, and this is a presentation on the development of a new tool for the identification of breast cancer patients at low to intermediate risk of recurrence of the disease post-chemotherapy. As breast cancer is a heterogeneous disease, the identification of patients who stand to have no benefit from chemotherapy is difficult. The proportion of patients in this category can be quite high. This represents a significant waste of clinical resources, along with generating unnecessary patient toxicity. This study looked at an alternative label-free biochemical imaging approach to imaging this tissue and developed an artificial intelligence algorithm to interpret the images and predict recurrence. The study used a tissue set from the University of Lund in which 142 patients with early-stage breast cancer received chemotherapy, 29 of which saw recurrence of the disease up to 10 years post-therapy. The tissue was imaged using our Fourier transform infrared microspectroscopy, and the images, which contain up to half a gig of biochemical data and are termed FTI or hypercubes, were then used as inputs to train a novel deep convolutional neural network, or DCNN, for prediction of recurrence. There are various examples of these networks available publicly. We developed a sample prototype which is similar in structure to LUNAT-5, and we compared its performance to similar networks and also to the gold standard genomic test called Oncotype DX, which is used routinely to segregate patients on probability of recurrence of breast disease after chemotherapy. The performance of all the networks was highly variable, which is not surprising given the small dataset which we used for training and testing here. However, we did find that our prototype DCNN did perform well and with a relatively small variance in performance, which is encouraging. And the performance of the assay is comparable to genomic and proteomic tests in this space. Ultimately, these results are quite encouraging, and we're delighted that Science Foundation Ireland are now funding the next steps in the development of the test towards validation and potentially clinical implementation. So all that remains is for me to thank colleagues at TU Dublin, and in particular Dr. Wei Ngayan, and my collaborators, Professors Liam Gallagher at UCD and Professor Karen Yearstrom at University of Lund, for collaboration on the study. Sincere thanks also to the Health Research Board and to Science Foundation Ireland for financially supporting this work. Finally, thanks to you for watching. We hope you enjoyed listening. Please do get in touch at tudublin.ie if you would like to know more.